So while we are studying enzymes, we, in the previous video, talked about what are enzymes, that they are usually proteins. We talked about the fact that they have a specific shape and that they can speed up specific reactions by bringing the substrate together in a specific way. So we want to go back to this diagram that was in the notes. When you're given this sort of diagram, one of the things that we want to notice is the shape of the enzyme, and we want to be able to identify the substrate. The substrate comes in, keeping in mind that we're probably going to see what's called induced fit, or a conformational change, that this enzyme will change shapes, and that is going to catalyze or speed up the reaction. Here we're breaking down sucrose into two products, glucose and fructose. And so we want to continue our discussion of enzymes by talking about two other properties. The first is that enzymes are reusable. If you think about something being reusable, it means it can perform its job again and again and again. And so the reason that enzymes are reusable is that enzymes are what we call unchanged during the reaction. RXN. And what we saw in the picture previously is that the enzyme's shape is unchanged. It can perform the same reaction again and again. We might see that slight conformational change, but once the products are released, the enzyme opens back up. And so what this means is that a single enzyme can do its job again and again. What we want to look at real quickly is this graph of enzyme concentration. So if we increase enzyme concentration, what happens, and we want to add in that this is the rate of the reaction, or the speed of the reaction. So when there is very little enzyme, the speed of the reaction is very slow as well. As we add more and more enzyme, then we find that the rate of the reaction doesn't increase exponentially or infinity. It actually begins to level off. So we want to think about, well, if the enzyme can do its job again and again and again, and I keep adding more enzyme, why wouldn't the rate of the reaction continuously increase? And the main reason is that the limiting factor, what's limiting this reaction, why it's not going faster, is actually then the concentration of the substrate. How much substrate do I have? So almost imagine being in a grocery store, and how many people are the cashiers checking people out? Well, if you have 100 cashiers, lots of enzyme, and only 2 people checking out, then the rate doesn't get any faster. Those people are going to check out just as fast if there were 2 cashiers and 2 people than if there were 100 cashiers and 2 customers. So here, the limiting factor becomes the concentration of the substrate. Because we know that as the reaction proceeds, the concentration of the substrate should decrease. The substrate's being consumed by the reaction. The concentration of the product is going to increase. But once I've used up, or once all of my substrate is being used, the reaction won't happen any faster. In fact, once all of the substrate is consumed, the reaction rate would drop again because the reaction isn't occurring. What isn't limiting is the enzyme. Once I have enough enzyme concentration, they're all working because they can continue to perform the same function. The fourth property that we want to look at is that the enzyme can be denatured. And we talked about the word denatured at the earlier part of the notes, because any protein can be denatured. We want to make sure that we can clearly define what it means to be denatured. That means that there is a change in the shape, and so it can no longer function. or it functions at a slower rate. It can't function as well. And we would want to be able to elaborate on what part of the shape for an enzyme as a protein is really the active site. If that is changed, then it's no longer going to react. And so there are a couple factors that we want to look at. The first is actually temperature. And temperature, somehow this got moved down. Temperature is really just a measure of molecule collisions or movement. And so what this has to do with is the idea that molecules are continuously in motion, or molecular kinetics. They're continuously moving around. And temperature is a measure of how quickly. So what is the speed of this molecular movement? And as these molecules continue to speed up, the reaction is going to happen faster. The faster the substrate is moving, the more likely it's going to run into the enzyme and perform the function. So if the temperature is really low, then the rate of the reaction is very slow. Once I hit my what's called optimum or optimal temperature, my rate is the highest. But if the temperature gets above that, if the molecules are moving too quickly, then the enzyme begins to become denatured. So we're not denatured when it's cold. We're just not moving fast enough. When it's hot, we want to remember that this change of shape is usually because bonds are broken in those 4 levels of protein folding. And so as the temperature increases, some of those bonds get broken, and that changes the shape of the enzyme, and the enzyme can no longer function. What we need to be very aware of is that different enzymes have different properties. So some enzymes are going to work very well at room temperature or the human body temperature. Other enzymes, for example, in bacteria that live in hot springs, they're not going to even work at room temperature. They won't work until their optimal temperature. So really, anytime we talk about a change in the environment, this is going to usually change the bonds. So the next thing that might influence this is also going to be pH, or the concentration of hydrogen ions. What we're looking at here are actually 3 different enzymes, pepsin and trypsin, and I believe this one is amylase. And what we can see is that as the pH changes, the rate of reaction or the effectiveness of the enzyme changes. Keeping in mind that pH is a measure of hydrogen ion concentration or hydroxide ion concentration, POH. And these are ions, and these ions can interfere with our bonding levels of folding. So remember 7 being neutral, so pepsin works very well in an acidic area. This actually makes sense because it's an enzyme found in the stomach. But trypsin does not work well where it's acidic. It works well where it's basic. It's actually basic in the small intestine where trypsin is. So what you need to be able to do, given a graph of enzyme reaction rates, is predict how the enzyme will function and explain why it would or would not function well. And then you could even design a lab to test whether this enzyme will have the predicted reaction rates. There are a few more things that we want to talk about that can influence the rate of the reaction. The first are called activators or enhancers, and they have a very similar effect. Basically, we just want to know that they would increase the rate of the reaction. And what these are going to do is they're usually going to bind to an area that is not the active site. And that's going to cause a conformational shape change. So bind to what we call an allosteric allosteric site. Allosteric is not the active site, so it's another place on the enzyme. When it binds to the allosteric site, we're going to see a conformational change or shape change. And this is going to increase the rate of the reaction. It might make the enzyme more likely to attract the substrate. And so the advantage of this is that we can have enzymes that are just kind of dormant or waiting, and then once something has arrived, then the reaction starts. So this is a great way to kind of regulate what reactions are happening in cells. Even though the substrate's here, nothing's happening until this other signaling molecule. So when we talk about hormones being able to signal to the cell what to do, this is one of the ways that it happens, is through hormones and other molecules being activators or enhancers. And so, of course, vice versa can happen. We can actually have molecules that act as inhibitors. Inhibitors are going to decrease the rate of the reaction. And there are two types of inhibitors. The first are called competitive inhibitors. Competitive inhibitors are going to compete for the active site. And so what this is going to mean is that they are usually similar in shape to the substrate, but no reaction will occur. So what we see here in this picture of competitive inhibition is that this molecule is able to fit into part of the active site, but no reaction is going to happen. And the substrate can't fit in. So we're decreasing the rate of the reaction because the substrate can't fit in. Sometimes these are permanent, and sometimes they are not permanent. So sometimes they will permanently bind to the enzyme and completely inactivate the enzyme. Sometimes they're just going to bounce in and bounce out. The second type are called non-competitive. And what we mean here is that they do not bind to the active site. But they bind to this idea of an allosteric site or an other site. And so we can see this picture here. Here we've got our enzyme. The allosteric inhibitor binds to the enzyme at a different place, and it causes a conformational change. Shape change. And now the enzyme does not fit the substrate. So here, the enzyme shape is intact. It's just that the substrate can't fit into the active site. Here, the enzyme shape has been changed or compromised, this conformational or shape-changing site. And so this is called non-competitive or allosteric inhibition. Again, these are ways to communicate to an enzyme, do this reaction, or don't do this reaction. Sometimes the body is using this on purpose. This is also a great way for pharmaceutical companies to target certain reactions. If they can develop a drug that will inhibit an enzyme, then they can actually control whether a reaction is happening or not. All of this comes into play when we talk about pathways of an enzyme, putting multiple enzymes together, and then being able to control what part of the pathway is working. And we'll talk more about pathways and feedback inhibition in the next section.